Okay, cool. That's the pilot.
Thank you very much, Robert. I think I talked in Imperial about, I don't know, just after you arrived here. How long have you been here?
Good question. Sort of 15, 17 years maybe?
Yeah, so about 16 years ago I talked here. Some of the audience have changed, I'm pleased to say. Yes, thank you very much. I was a programmer and I got sick of it, so I became a biologist. And then after some years as a PI, the gene sort of activated itself again, and I flipped back. Very difficult to do. One of the things you don't realize is how much the difference between computing field and cell biology field and the math field are actually to do with the behavior patterns and thought patterns of people in those fields. And it's actually very, very hard to jump from one to the other, and it's been great fun doing it. I've learned a whole load. Anyhow, here's a bit of a nice introduction to what the problem is. You all and I started like that. You all and I ended up like that. Now, in order to get from the one to the other, obviously, you need a huge increase in complexity. We no longer believe that there's a little human physically encoded in all of that. All the information must be created by the cells themselves. In order to do that, you must have a great increase in the amount of information. And when you've got all that information, or as part of getting that information, you need the different cells and layers of cells to move with regard to one another, or you'd just be spherical. These are the things I'm interested in. These are the things I want to understand. This puts the work my group does quite a lot apart from the chemotaxis field, because very often what the chemotaxis field wants to study is how cells chemotax along a pre-existing gradient. And the whole point about this is there can't be all the pre-existing gradients there. What's interesting is as much where the gradients come from and how they're made as how the cells respond to them. Just as an aside, because it's fun, my twin daughters have been staying with me this last week. One of them is into politics, so we were watching the election last night, and if I suddenly fall asleep on you, then you'll know what the problem is. I saw my daughters as 4-cell embryos. They're test tube babies because we were academics of a certain age. And the first round of test tube babies we had, I said, "Can I see, please, because I'd like to." And they rushed me out of the place and said, "This is illegal, this is horrible, get out of here." And the second round we had, we said, "I'm a professional microscopist, and I really don't think the gear you've got is the right stuff." And they had me in there like a shot looking at. So I saw my daughters when they were 4 cells. Giving thanks first. Luke, you know here, he was a PhD student with Robert. Andy is a medic. They're winning a prize for the best medic PhD. John is a mathematician. Michele is an engineer. Adam, a mathematician. That really expresses how we all are. We're a complete mix of maths and physics and biology and medicine. It's great. And a whole load of other people. You all for listening. I'm going to do a bunch of newish immune cell stuff, and the immune people are there. Right. What am I going to talk about? Self-steering, mostly. I'm going to talk about how cells break down chemoattractants to steer themselves. I'm going to talk about how cells make chemoattractants to steer themselves, and I'm going to talk about conclusions coming from that. I discovered when I was tidying up this talk that there was just not enough time to talk about reverse chemotaxis. If you are interested in reverse chemotaxis and you want me to discuss it, please find me afterwards or email me or such like. I'm sorry, I know I put it in the abstract, but I can't go too far over time. Okay. One good place to begin this talk, melanoma. Melanomas spread by chemotaxis. You are very, very frightened if you have a melanoma. You're very, very frightened not because a black spot on your skin is a problem, but because you are certain that if you have a melanoma for very long, that it will spread. There are no patients with big melanomas. because the patients are dead before the melanomas are able to become big. And when you look at a melanoma, you can see that it's spreading, because if you look at the edge of it, some of it looks a little bit like a Gaussian, but some of it doesn't look like a Gaussian at all. And if the cells were just dividing and spreading out randomly, then you'd have a circular, probably a spherical Gaussian. But this kind of unevenness, raggediness, it's the definition of a melanoma. That's what doctors look like. And what it's actually telling you is parts of this tumor are chemotaxing out of the tumor and into the rest of you. And in an assay we did earlier in the Beatson, you can see this happening. This is a chemotaxis device. We have a pool of high serum medium there and a pool of low serum medium there. And you can see the melanoma cells chemotax outstandingly well from the low serum to the high serum. And when you see this, you should become very frightened if you understand what it's showing, because if this was a dark spot on your skin and the cells were doing this, then the melanoma would be going to kill you. You're watching kind of the process of a melanoma killing its patient in capsule here. The question we asked, as I've sort of warned you in the first slide, was not by what means is the melanoma actually reading the gradient and migrating up it. I don't think that's terribly interesting. I was asking a different question, which is, where does the information come from? You can't spread from a melanoma to outside a melanoma unless you know where the melanoma is and where outside is, and it doesn't seem obvious that a cell should know that. So the chemotactic gradient, if it exists, must be low inside the melanoma and high outside the melanoma. And you have to ask, why would your body generate a chemotactic gradient in order to get cells out of melanoma and kill you? It seems a bit stupid. But actually, of course, the answer is your body doesn't generate a gradient to take the cells out of the melanoma. The melanoma cells themselves generate a gradient that takes the cells out of the melanoma. So the information is a chemotactic gradient, but that information is being made by the melanoma cells. Best way of showing this was by doing a control. And as a result of experiments like this, we have a lab rule that you celebrate cool results twice. First, before you do the control, and second, after you've done the control, if it worked, because so often the controls tell you weird stuff. So here's the control that should not have worked. We have high serum here, and we have high serum there. There is no gradient. If there's no gradient, the cells should not chemotax. But what do the cells go and do? They move exactly the same as when we imposed a gradient on them. And just interpreting many, many years of work and so forth, what's going on here is there is an attractant in serum. It's LPA, lysophosphatidic acid. And the gradient here is LPA, but it's set up asymmetrically. It's set up with cells only in one place, and cells are incredibly aggressive at using an enzyme on their surfaces to break down LPA. So wherever the cells are, there is no LPA left. Wherever the cells aren't, there is still some LPA, and diffusion sets up a gradient between them. So the gradient is being made by an interaction between the cancer cells themselves and their environment. And you can check all due controls. So if you look at melanoma cells grown without LPA, they're not making any. But if you put a low density of melanoma cells in a culture that does have LPA, they break it down. And if you double the density of melanoma cells, then, sorry, it's 8 times this one. I'm reading them wrong. If you have a higher density of melanoma cells, they break down the attractant faster. So it's all pretty well behaved. So what we think is happening when a melanoma in you spreads is the melanoma is there, it's growing, it breaks down a gradient of something that is produced by your body. And when the melanoma becomes big enough to establish a local gradient that can't be replenished by more stuff diffusing in, then suddenly at that point, the cells of the melanoma perceive a gradient and will start migrating out. And that is why there's a magic thickness of melanomas. If you have a melanoma that is thinner than a certain amount, your physician will say, your oncologist will say, oh, don't worry about it, we'll cut it out and you'll be fine. And if it's thicker than a certain amount, the oncologist will say, oh, it has almost certainly spread, you should have chemotherapy. And the difference is just that when the melanoma is small, when it breaks down chemoattractants, then there's a small amount of tumor and a lot of tissue, and more chemoattractant can diffuse in and there's no gradient set up. But there is a critical size, and when the melanoma reaches that critical size, then you start getting a gradient, and then the cancer cells start spreading. That's a quick tour through it, but what we want to do is get a mathematical and modeling understanding of what's going on. So if I just start by illustrating this with a toy model. For the biologists here, toy does not mean it's a silly plaything. It means we have not made it match reality. We've just generated it to show what's going on. So here you see lots of attractant shown in red. And here you show, as I showed in my earlier movies, the cells start in one place. And the cells break down the attractant. As the cells break down the attractant, it sets up a local gradient. The cells walk along the local gradient. The whole system feeds back on itself, and you get a wave of cells migrate out by chemotaxis to a molecule that they are breaking down. So this system has lots of attractant in it, much more attractant than you would think if the cells were just responding. And it's actually very telling if you go and look through all of the literature about all the chemotaxis experiments that people do. They nearly always add much too much chemoattractant for the experiment they think they're doing, which is giving the cells a gradient. What they actually do is giving the cells an attractant that the cells can break down until the cells make a gradient. And again, just to keep hammering this point home, this gradient is self-organized. The cells create the gradient while they are moving up it. They don't create the attractant in this scenario, but they do create the gradient of the attractant, and it's the gradient that has the information in it. So we keep seeing this system happening again and again and again. We keep seeing that cells prefer to break down an attractant that comes from outside rather than making an attractant themselves. Why should that be? Hans Meinhardt, who's one of the pioneers in the field of theoretical biology and gradients, said that secretion of a substance that's repellent is exactly the same as breaking down an attractant. And the thing is, that's true if you're a mathematician and these substances are continuous. But if you have a substance that is detected by receptors, it's not true anymore because receptors saturate. So the reason that it works really well if the cells are breaking down an attractant that comes from outside goes like this. This is an experiment we had to do as because a referee demanded it, and it was a wonderful experiment, and I'm glad the referee demanded it. So this is cells doing a self-generated gradient over about a day. So the scale of this is much larger than you've generally done. And you can see again you have this consistent wave. But the wave stays consistent however far the cells go. And the wave stays consistent because, let's come back, start that again. Because the cells that get ahead of the leading edge become saturated. And they don't experience a gradient anymore, so they stop migrating in a directed way. Whereas the cells that are just a little bit behind the front of the gradient, they're seeing a very steep gradient at the tail of the wave, and so they go a little bit faster than the speed of the wave. So this wave of cells migrating is not determined by the cell speed or the properties of the chemoattractant gradient. It's determined by the interaction, and it's self-sharpening. And you can maintain a wave like that for ages, and it's very, very robust. This robustness gives you some interesting things. So if you compare a model of cells chemotaxing to an imposed gradient here, the blue is where the gradient's coming from, the gradient is shown in colours, and the modelled cells are there. The cells that respond to a gradient that you give them start off like mad. But then, as they get further and further up the gradient, they get more and more saturated. And also, the difference between their fronts and their backs proportionately gets lower and lower. Obviously, when they start, let's look at that again, it's nice. When they start, there's nothing at their backs, and there's something in their fronts, so there's an absolute difference. But the higher they get up this gradient, the less difference there is between the receptors occupied at the front and back, so they slow down and they get less good. The self-generated gradient has no such problem. Wherever it goes, behind the self there's no attractant, and in front of the self there's some attractant. So you have this very, very strong local gradient that the cells carry around with them, and it can go on forever. So you've got something that is far more robust. It can deal with a far wider range of concentrations of attractant, and it can deal with complicated shapes. It can deal with circular shapes with cells growing outwards, so on and so forth. If you go to a really big gradient like this one, this is modeling, we're now parameterized, and we're now modeling a 2 millimeter gradient. Again, the same thing happens with the imposed gradient, but the cells get less and less good at reading it as they get more and more saturated, and as the gradient gets flatter and flatter where they are, and they actually never reach the end. But the self-generated gradient will just go on and on and on. It doesn't make any difference. So actually you can conclude, if you look in an embryo, or if you look in a cancer, or if you look in any situation where chemotaxis is supposed to be happening biologically, you can conclude that if it's less than 0.5 millimeter, it could possibly work with an imposed gradient, but if it's more than 0.5 millimeter, it just couldn't. There's no way you can tweak the saturation of the receptors and the shallowness of gradients and so forth. You can only chemotax more than something like 0.5 millimeter if the cells join in and take part in making their own gradients around themselves. This is just, showing reality again. The top shows the models, the bottom shows some real cells, and what we're doing here is using a chemoattractant that cells can't break down and a chemoattractant that cells can break down. And you can see the self-generated one starts off exactly the same, but then it carries on going just as the cells with the unbreakable gradient start to run out of energy. Robert?
So what if you had now a sort of a circular front, sort of in 2D? Then I guess if you have a fixed numbers of cells, they would run out of cells to have a coherent front, and it would get more and more diluted, essentially.
Yeah.
Does this then stop in some sense?
Yes. No, it's a very, very good question. So if the real problem with this is that if your front expands, then you have fewer and fewer cells per unit width of the front, and eventually they're not going to be able to set up a new gradient. But one of the things, there's a bunch of slides of this, but I took them out because there wasn't time. I won't dive into them. But one of the things we discovered quite early on is that most of the chemoattractants that are involved in embryogenesis, as well as being chemoattractants, are also mitogens. And this has actually stopped people understanding most chemotaxis during embryogenesis, because anything that's both a mitogen and a chemoattractant is going to be quite hard to work in. But you imagine if you have a small front of cells that's growing out in 2 dimensions into a big front, then it's going to run out of energy. But at the point where it runs out of ability to make a gradient, all the cells are going to be in a high concentration of the attractant. And so if it's a mitogen as well, then it'll cause them to divide until there's enough of them to break down all the attractant, and the whole thing is self-sustaining. Luke showed that. Again, there is a wonderful habit of biology to make these very, very robust systems, and that is just a nice way of doing it. So where might you expect to see this sort of self-generated gradient? I've talked about melanoma, but here is a case in a developing mouse. So developing mammals are unpigmented. Embryos are white. But at a certain point in embryogenesis, the melanoblasts come out of the neural crest and migrate through the skin round from the neural crest round to your tummy, from the neural crest down your shoulders and down your arms. Doesn't matter whether you're a mouse or a bird or you, it works pretty much the same way. And people have really struggled to understand how that's possible. And as I've said, the distance the cells have to migrate when they're going from the neural crest round the body and down the developing arm is greater than 0.5 millimeter. So it can't just be to a gradient. And lo and behold, in this glorious piece of research done by a postdoc called Olivia Susanto, where she's looking in a still living ex vivo mouse embryo and looking at GFP melanoblasts migrating down the arm, you can actually see exactly what we would predict, which is that the cells in the front 300 microns or so of the wave of melanoblasts are moving in a highly directed way, whereas the cells behind are moving in a totally undirected way. And we infer, but we haven't been able to measure it, that these cells are making a self-generated gradient that is driving the cells from the place where the cells are to the place where the cells are not. But in doing that, the front wave is consuming all of the attractant, and everyone behind has no information, just as in the model I was showing you before. So this is actually quite a nice explanation for mouse pigmentation, for mammalian pigmentation. And the wonderful thing about this is while we were doing these models, my wife, who is also a cell migration biologist, but much more cancery and much more involved in things like this, was getting indecently interested in the phenotypes of mouth that had a white belly and a white forehead. Because it turns out if you have a motility problem in your melanoblasts, then that's what you get. They just migrate a little bit more slowly around. And it turns out that you can model the whole process of mice acquiring pigment in a way that explains pretty much all of the known pigmentation mutants by just combining a couple of self-generated gradients, one of endothelin 3 and one of KIT. So I'll just show you, I haven't time to explain it all, I just thought you'd like this. There are cells, they're going to start breaking down the endothelin 3, and when they start breaking it down, they'll make a gradient that moves them out. This is in the dermis, and when they run out of endothelin 3, they jump into the epidermis and do the same thing with a gradient of KIT ligand. And Robert, you will see that the cells are responding to both of these things by dividing as well as so that the interface is stable. And you can see that the last place that the melanoblasts arrive is in a little patch around your tummy button, so this fits the mutants perfectly. And you can go through all the mutants you know in pigmentation, and you can pretty much map them all into a change in 1 or 2 parameters in this model. This is the one we were particularly proud of. This is called the down under mutation, and it's a mouse that just forms pigment right here in the end place where these cells have migrated. And it turns out if you go and sequence the mutants, that they produce much less of the endothelin 3. So there's nothing to sustain the cells that are left behind the leading edge. So the leading edge moves forward in exactly the same way, but it's not able to leave anyone behind it. And if you put the same thing into the model, then you exactly recreate the phenotype of the mouse. Isn't that nice? Sorry to give you such a brief introduction to that, but I thought you'd like it, and I can always tell you more about it later. How are we going to measure all of these things? So the trouble is, and this is why everyone doesn't know about self-generated gradients. This is why everyone doesn't think about self-generated gradients. They are really very, very hard to measure. If you have a little local patch of cells and they're breaking down chemoattractant and new chemoattractant is diffusing in, you can't go and take a big explant and work it up and sequence it or do biochemistry on it, because the cells have to be very, very rapidly breaking down the attractant, and new attractant has to be diffusing in. So these models that I'm showing you are actually the only way at the moment that we can make an effective way of understanding and putting parameters to a whole load of the properties of this. But the models are great. The models fit beautifully well with what we see in biology. And what we like to do is we like to go and study the biology quite a lot and spend quite a lot of time measuring the things that will go in to make the parameters of the models. And then we put them together in a model. And then the model doesn't look anything at all like what happens in reality. But then you go and say, you stop and you think and you say, what have we got to change? And you either change the biology or the model until you get it right. So that lets us do some really cool things. So we built a model that we were pretty certain recapitulated reality. And once we'd built that model, we started toying with it because we had lots of ideas. We asked ourselves, what happens if you get a bunch of cells doing a self-generated gradient and going along a fork? And the answer is a simple fork. The cells split 50-50 either way. If they were moving up a fixed gradient, then randomly each one of them would either go to the left or the right. So you get quite a lot of randomness between how many go to the top and bottom. But in a self-generated gradient, the number of cells determines how far the attractant is broken down. And so you actually get a precise split between the cells that go into the two halves of the Y junction. But if you go and make it more complicated and connect one arm of the Y junction to the outside world, so there's more chemoattractant coming in, but you leave one arm as a blind end, then what happens if the cells will migrate up to the start of the branch, and they'll split both ways, but then the ones that have gone up this way break down all the attractant, so subsequent ones go down the other way, so you get a sort of asymmetric response. Now what we ended up thinking though is, hang on, there's a more interesting scenario. What happens if the blind end is short? What's going to happen then is that as these cells near the junction, they break down the attractant diffuses out of the blind end, but by the time the cells have to make a decision, then they'll have broken down lots of attractant from here, and there will be less attractant in the blind end and more in the connected end. And so they'll be able to make a decision which way they should go without ever going down the wrong way. They'll be able to see around corners. And you can see that happening in this model. And if it works on a nice, simple Y junction like that, you can start building arbitrarily complex things like mazes, like this. And what you see, if the cells create a gradient, each time they arrive at a branch, each time they arrive at a branch point, then they've broken down more attractant in the wrong place, less in the right choice, so they can go all the way through the maze, making correct decisions, even though the decisions are around corners and they can't see the ends. Now, this is absolutely glorious, but of course it's just a computational model. But the thing is, over the years, we refined our computational models to the point where actually they were basically right. And it turns out if you build a device to look like this, then real cells do exactly what the models say. The real cells will find their way through mazes of arbitrary complexity without ever going down the wrong branches or without ever needing to go around the wrong branches. They really can see around corners by using chemoattractant breakdown and self-generated gradients. Isn't that fantastic? And you can, as I say, you can make the decisions more and more complex. Here we have the Hampton Court maze. Some of you will recognize it. Any of you been in Hampton Court maze? The people make a really bad fist of Hampton Court maze. The cells do much better because they can see around corners. People can't. Now look at an interesting thing here. There's a glitch in the maze. Let's just start it again. There's a glitch in the maze. And the point is these cells are not learning the rules of the maze. They're just exploring their environment and finding where the best path to the chemoattractant is. So if there's a glitch in the maze, the cells can tell it's there by breaking down chemoattractants. And this reminded us a lot of IKEA. So a few of you have probably done Hampton Court Maze. I think probably nearly all of you are unlucky enough to have done IKEA. But there is a secret in IKEA, which is they have little hidden doors. And if you can find the little hidden door, then you can avoid the entire sofa section. And avoiding the whole sofa section means you save an hour and a half looking at crap sofas, and you get much less unhappy, and you live for an hour and a half longer, and it's very, very good. So in our Hampton Court Maze, we put an IKEA back door there, made a new maze, and wondered if the cells could respond to it. And what happens is actually really both very, very funny and quite sad. So the funny thing is, yeah, they saw the way straight through. The sad thing is now, I've shown you models of this, you know what's happening. These cells are breaking down all the chemoattractant that's diffusing in from here. So the local gradient is here, and there's no chemoattractant left here. So all these cells have no information. They will never escape the maze. So some cells found the IKEA side door, but in doing so, they doomed everyone else to the rest of their life in the sofa section. What this all emphasizes is that if you break down chemoattractants and chemotax to the resulting gradients, what you can actually do is you can interact with your environment and test out your environment at really quite significant distances and in environments of quite significant complexity, and you can understand your environment before you need to go into it. So the conclusion is not just about making chemoattractant gradients, but is something much more general, that by breaking down attractants and letting them diffuse and so on and so forth, if it is tuned correctly, you can use this to get amazing information about the environment and about what the source of the attractant is and where it is, that you would never be able to get normally. And we love this. Just a little aside, because Robert's here and would like this, how good are our models? So when you look at this model that I showed you, you see, again, that there is a small group of cells that has all the information, breaks down all the attractant, and everyone else is lost. If you look at the thing I showed you here, I said it was identical, but it's not. Group of cells at the front, but the cells behind are all following them. The cells behind do have some information. We noticed this and we said, why are our results wrong? And it turns out that these are dictyostelium cells, not that it matters much, but they're dictyostelium cells. And dictyostelium cells talk to one another, and they talk to one another using adenyl cyclase, and you can block this by adding caffeine. But what we discovered is, at the tiny, tiny scale, these branches are about a nanoliter or that sort of quantity, that the caffeine doesn't work well enough. So in order to test whether or not our models were really right or not, we deleted the gene for the adenyl cyclase that makes the cyclic AMP. And when you delete the gene and repeat the experiment, you find, yes, the cells behave exactly the way that the model predicts they would. So we've reached this wonderful place, which was reached by not very many modelers, but is wonderful when it happens, where if there is disagreement between biology and the model, it's as likely as not biology that's wrong. Dennis Bray got it with bacterial chemotaxis. They found some amazing things about bacterial chemotaxis, which have defined the biological field since because of disagreements between the models and reality. This was not a huge surprise, but it's absolutely lovely to see. So, models are great if they change experiments, but I would argue to wet biologists that are here that experiments are only great if you can make a model from your experiment, and if the experiment tells you how to change your model to make it better. And we discover stuff when the models improve the experiments, and the experiments improve the models, and so forth in a circle. The cycle is the key. Okay. I've shown you models and I've shown you dictyostelium. How widely in biology would you expect to see self-generated gradients? We have been looking at this for about 10 years now, and the conclusion is it's absolutely universal. We have never seen a case where chemotaxis happens in a real living system where the cells don't at least change the gradients to suit their own selves. We put some cancer cells in our mazes. These are, I think, melanomas, but they might be pancreatic cancers. I've forgotten, I'm afraid. They're very similar in their behavior. But the cancer cells find their way through the mazes by chemotaxis. They get a little bit lost when the mazes get complicated, which I'll talk a little bit more about later. So cancer cells, just like dictyostelium, create gradients to find their way out of steering problems. Recently, we've been looking at macrophages. Now, these are bone marrow-derived macrophages from mice. We found we can actually get basically physiologically good macrophages from HOXB8 cells. So increasingly, we're using those. But anyhow, these are good, well-behaved macrophages. This is the same experiment I showed you before. So there's a bridge, there's a well below, and the macrophages are put into one well. And in this system, there's no C5a, macrophage chemoattractant. And in this system, there's 3 nanomolar C5a everywhere. So no gradient, no information unless the cells make it themselves. And what do? That's great. Nobody has got macrophages to chemotax this accurately in another experiment. They're just perfect. And if you go and look at the dose response, so if you add different amounts of the chemoattractant everywhere, you see exactly what you would expect to see, which is to say when you have a tiny amount, 0.3 nanomolar, a few cells come out. 10 times the amount, you get lots more cells coming out. 100 times the amount, you're going to get a wodge of cells in a wave, like I was showing you before. This is what you expect, because your wave of cells at the front has to be enough, but no more than enough, to break down all the chemoattractant that's diffusing in. So you expect to see a match between the number of cells migrating downwards and the concentration of chemoattractant migrating in. The other thing, again, you see, and this is going to be a theme, so I'm going to have to segue to this, is you see a wave of cells at the front moving out to an obviously self-generated gradient, and then you see a bit of a pause in this case, and then you see a whole load of other cells following them. And by analogy with the experiment I showed you before, we're pretty sure that macrophages in front must be talking to the macrophages behind and telling them to come too. And it must be through a chemoattractant as well. And that's just the quantification showing it is just as we expect. The number of cells goes up as you add more and more attractant. And here's a very interesting thing. I know there's some immunologists present. If you read the literature about macrophages and C5a, there's lots of description of an enzyme called carboxypeptidase M, where M stands for macrophages. And carboxypeptidase M is a cell surface enzyme that breaks down C5a, makes it much less active. So we presumed totally that what was going on in these glorious experiments was carboxypeptidase M was breaking down the C5a in exactly the same way that the enzymes were breaking down the LPA and the cyclic AMP. But it turns out that mouse macrophages don't have macrophage carboxypeptidase. It's a human-specific phenomenon. Carboxypeptidase M is basically not expressed in mouse macrophages. So what the ones we're looking at aren't doing is they're actually endocytosing the C5a. This is an experiment with fluorescent C5a, and you can actually see them live creating a gradient as they migrate. You can't quite see that it's happening here, but you can measure it, and I'll show you a more visible one later. So the cells are taking up C5a from the medium, using their receptors and destroying it. And by doing that, they're creating a local gradient that they can migrate up. And if you look at them with a bit more magnification, you can see that the place that the fluorescent C5a has gone is all into all the lysosomes and endocytic vesicles of the cells at the front of the chemotactic wave. This one you can see, it's a little bit clearer between the top and the bottom. Actually, it's clearer on my computer screen than this, but you can see it happening here. This is just an interesting thing that we have observed regularly, that when you have cells migrating up a linear gradient, here we're giving them no C5a to some C5a, by breaking down the chemoattractant, they turn a linear gradient, which is hard to respond to, as I showed you earlier, into an exponential gradient, which is much easier to respond to because you have a constant ratio between how many receptors are occupied in the front and in the back. So yet again, the cells are actually improving the quality of the information they're able to get out of the environment by breaking down a chemoattractant. And we can see it happening live with this fluorescent C5a. Exactly the same story with dendritic cells. These are dendritic cells chemotaxing to CCL19, and they do an absolutely amazing job of finding their way through a maze. CCL19 is the chemokine that takes dendritic cells from an infection to the lymph node. And you can imagine that this is actually the same problem that dendritic cells normally face when they're in your body responding to an infection, and so it's not surprising that they do well. But again, look, lots of cells follow the front ones. It's not just a glom of cells at the front and then no information. So again, we are pretty certain that the dendritic cells at the front are sending their own chemoattractants to the dendritic cells behind. And you can actually see it happening here. Look, at first, only cells go down this way. At one point, one cell randomly migrates down the wrong way. And once a cell has randomly migrated down the wrong way, then all its friends cram in after it. So that first cell going down there sent a message, like an ant. Actually, we think the ant is probably a good illusion. And so on, and so on, and so on. We think that everywhere where you see chemoattractants, that the cells are breaking down the chemoattractants and making their own information. But the extra bit leads me on to this last section that I want to talk about, which is what about cells making their own attractants? And I know about this from my PhD and my postdoc. Dictyostelium cells, that's the thing that dictyostelium cells do. What you see when you look at dictyostelium cells is one cell there starts generating a chemoattractant, and its next cell along chemotaxis in towards the first one, and then releases a pulse of chemoattractant itself. So the chemoattractant, which is cyclic AMP in these cells, goes out in a wave, and it goes out in a constant self-generating, self-sustaining wave until all the cells pile into that one cell at the middle that started it all. Now, for ages and ages, if you've shown movies like this, the wise medics have sat in the front row and said, yes, but dictyostelium. And then last year, this amazing paper by Strickland and Weiner comes out, showing that if you spread neutrophils on a glass dish, and you give them a totally insoluble, non-diffusible stimulus, which is just yeast, then the neutrophils do exactly the same thing. Except that the neutrophils never start making chemoattractant until they hit upon something they want to chemotax towards. I'll just show the magnified one, because this is gobsmacking. We've been watching neutrophils chemotax for so long, and we never saw anything like this. You're looking at a calcium probe here just to show if the cells are signaling. And you see that they jump in, and then they secrete something that makes their neighbors go in. That something is leukotriene B4, and it's amazing. The key points, though, and even the authors of this paper don't emphasize the key points enough, is that all of the cells chemotax towards the yeast, but the chemoattractant is not made by the yeast. All of the cells chemotax towards the yeast, but the information comes from the yeast. The chemoattractant comes from the cells themselves that they're making on the fly while they're chemotaxed. So it's the difference between information and gradients and attractants yet again, but it's cool. So the immune response when your neutrophils are responding to something like this is a collective phenomenon. One immune cell is not much use. A bunch of immune cells can talk amongst themselves and decide how to fix you. So my rather aggressive theory is that actually there isn't single-cell chemotaxis in vivo. If you take a single cell and you put it in a device, it can chemotax. But my belief now is that any time you see cells chemotaxing, they respond to an attractant, but they also talk amongst their friends with chemoattractants, and the whole lot of them migrate together. Why do I think this? Because in all the cases where we can actually see stuff microscopically and understand it, like schools of fish or hives of bees or so on and so forth, ants, termites, you don't see a single creature acting on their own. You see a society of creatures making information collectively about their environment. I'm running out of time, so I'm not going to talk about this, except to say there's no conceptual difference. The idea is the same. So we make models. We make models where the cells secrete for themselves an autoattractant. But the trouble with secreting an autoattractant is with that alone, the cells will just all do the dictyostelium thing and pile into the same spot. So we model the cells also secreting a shorter range repellent. And then you get these rather nice societies of cells that migrate around. And if you look at how these modeled societies of cells respond to an external chemoattractant, here you see a group of modeled individual cells responding to this chemoattractant here in blue. Here you see the society of cells responding to the chemoattractant. And you see that the society of cells does a much better job at responding to the gradient of chemoattractant than the individual ones can. So I think this is what your immune system looks like. This is what everyone's believing at the moment, but this is reality. And if you block leukotriene B4, which was the swarming hormone for neutrophils, you find that this second wave, sorry, this is a very fussy slide, but if you compare these two levels here, 10 nanomolar C5a and the same with something that blocks C5a, you still get a wave of cells bounded by this red box moving out. But if you block LTB4, then no one comes behind. So one of the chief things that macrophages seem to be using to bring all their friends is that they're secreting LTB4. And actually it turns out that even when you just do a normal chemotaxis assay, if you block LTB4, the cells chemotax a whole load less well. So even when you would think that all the cells are able to respond on their own, they don't. It's much better if you can talk to your friends. And I showed you that. And we don't know what the chemoattractant is that the dendritic cells are using. We're working on it. Come and join us, please. We could always deal with immunologists who want to solve lovely problems. Okay, so the final conclusions, because I'm running short of time. All of the single-cell chemotaxis conditions that we think we've studied aren't. You can study chemotaxis in single cells, but what's going on in your bodies and in any real situation is an interaction between different cells of the same class, and probably in the case of immune cells, different classes of immune cells as well, all responding to signals from one another and sending out signals to bring one another in. Collective migration is the way to go. There are loads of different mechanisms. So when the cells are breaking down the chemoattractant, they all move together in a front because the chemoattractant is a shared resource and they're all breaking it down. But if you break down the chemoattractant, you break it down for your neighbors. So that gives you collective migration. The dendritic cells and the macrophages are secreting a chemoattractant. It looks completely different, but it's the same idea. The cells move together as a society. And finally, it will, I'm sure, turn out that secondary attractants are very, very common. So when a cell sees one chemoattractant, it secretes more of that attractant or a different chemoattractant or an enzyme that breaks down chemoattractants. And I'm going to leave you with a final hypothesis, which is that there is no difference between immune cells chemotaxing and fish schooling. The fish have the advantage of sight. They can see what everyone else is doing. The immune cells are basically chemotaxis factories. They're covered with receptors for chemokines and lipids and things so that they can smell what their neighbors are doing. My hypothesis is that it's exactly the same apart from the details. And I'll leave you with that. Thank you very much.
We can talk about this paper here. Very nice.
Yeah, I'm supposed to go blank.
Yeah.
I have to ask this question.
Yes.
So, real cells in tissues,
Right.
many have to go through stroma as well.
Yes.
How does that work? I mean,
Well, so all of...
usable
Yes.
attractant, I can see
Yeah.
they have to also break down stroma, right? To through them, or?
I would not want to say that cells moving collectively excludes all the other things that cells do. All the other things that people have observed are surely there. So if a cell is trying to get through stroma and the stroma is obstructing it, which happens sometimes, not perhaps as universally as people say. We were worried about it in the melanoma for ages, and it turns out the melanoma doesn't care. That the stroma's gone long before the melanoma starts migrating. And lymph nodes are fairly squishy. I don't think you need proteases to get into the cortex of a lymph node. But in those cases where you need enzymes to get into a tissue, you need enzymes. There is a really interesting thing, though. Everyone knew from about 1990, or maybe even 1985, that matrix metalloproteinases also chop a bit off most chemokines. So if you express MMPs on your surface, like MT1 MMP, it will help you get in through the stroma and break your way through the collagen, and it will modulate every chemokine that comes towards you, usually in a predictable way by trimming a little bit off the front that changes its affinity. So actually, probably using proteases to make a hole in your environment and using proteases to break down chemokines, probably the cells are saving a bit of energy by using the same proteins for both.